So we start with the sequencing reads which come from those sequencers, and our objective is to put these reads together into the chromosomes, and from those chromosomes we can assemble a genome. So this process is called as genome assembly. We define genome assembly as the process of reconstruction of original chromosomes based on overlaps among those short reads, and this process assembles those reads together into what we label as contigs, and contigs then assemble into those scaffolds. So here is an overview. Again, we can see that we get large number of those short reads which are coming from sequencers. One approach is we can put them together and we align them with one another and see their overlap. So that process is called as de novo assembly. The other process is we can see guidance from already assembled genome. We call it as reference genome. So we take the reference genome and we align these reads onto that. So that is simply the alignment. And this kind of assembly is called as resequencing. But if we compare both of them, de novo assembly is kind of a cumbersome or a hard process. Why? Because we are using those short sequencing reads and we need to put so many reads together. They are really small size. Why we are using those short sequencing reads? Let's look into this picture. So here we have a comparison between typical Sanger method. We are comparing into three different matrices. Number one is how many MBs, million base pairs we can produce a day. Number two is we are also looking into their cost, and we are also looking the read length which we are getting. So here is what we have in the comparison. So Sanger can give 1 MB per day at the cost of $400, and the read length is longer, which is 650, whereas 454 gives 1000 MBs in $15, whereas the reads, their length is 450 base pairs. Here we have another big picture where we have like Illumina HiSeq is producing a very cheap per MB cost, that is $0.1 per MB, and it's giving us those short reads and it's producing 20,000 MB per day. So we have huge amount of data, we have huge throughput, and we have the cost is really low. So this is the reason why we are using these short sequencing reads, guys. Let's look into some terms which we will be using in these assembly lectures. One is contig. So contig is a consensus sequence of DNA that has been assembled from overlapping DNA fragments, or what we can call them reads. Scaffolds is when we put these contigs together, then they become scaffolds. Here we see two contigs, contig 1 and contig 2, and in between there is an unknown sequences, so there is a long runs of ends. So these ends, we don't know what nucleotides are there. So this is where the contigs, they actually break. So we have those reads which are aligned together, and then there becomes some gaps. So these gaps are actually among these contigs. So when we put those contigs together, that thing becomes a scaffold. A captured gap is a gap within a scaffold. The order and orientation of the contigs is known. Here we see it again. So we have those contigs up there. So contigs are put together and they are put together into this whole thing is a scaffold. And there are between those contigs, we have those gaps, as we have seen there, large number of ends in between. So these are the captured gaps. Here at the top, you see these small reads which are linked with one another with these lines, or what we call them as edges. Actually, these are paired end reads. So we will cover them in a moment. One read is in one of the contig and the other read is in another contig. So this pairing actually helps in determining the right orientation of those contigs within the scaffolds. So misassemblies are common. What misassembly is, like, we can observe something where we have genome is incorrectly assembled, or we can have some places where we can have sequences are deleted and the neighboring segments are then crunched together. So these kind of issues, they are called as misassemblies. So we'll talk about them later. Here is the genome assembly as a whole, put it together. So we start with those short reads, and then the reads are put into contigs, and then contigs are then arranged into scaffolds. So there are different assembly algorithms which we are actually using. Common examples are all paths, velvet, miraculous. We'll talk about them later. So in the end, what so far we have seen here is that assembly is the process where we put these reads together, and we might see some misassemblies also.